What going on all here? Biology paper 2 broad topics review. Here we go. But first, a message from our sponsor.
Let's play
Raid Shadow Legends.
Start.
You're raiding thing. You're doing thing. You're doing thing. You're downloading thing. You're raiding thing. In the shadows. Raid Shadow Legends. So we're going to review past papers to secure that pass in CSEC. But you know what else you want to secure? Your data. So hook up with NordVPN.
Now, before we get further, I'd like to thank our sponsor, NordVPN, allows you to price and
So we're looking at some growth and germination questions first. So here we are on June 2017, question 1. Everything done answered here, so you just have to follow along. You're getting that one real easy in biology. Alright, let me look at question 1A. In an experiment, some students put corn seeds into each of the two test tubes labeled A and B. And you can see A have a temperature at 20 Celsius and B have one at 10 degrees Celsius. Write a suitable aim for the experiment. But you notice that both of them have moist cotton wool. So what you want to do here is to investigate, well, what is the manipulated variable? In that case here, the answer is temperature. So to investigate the effect of temperature on germination. In which of the tubes A or B would germination most likely occur? Well, you know you need several things for germination to occur. You need oxygen, the thing need a food storage in the endosperm or the cotyledons. You need a reasonable temperature and you need water. So in this case here, both of them have water, but what you want to look at is the temperature. So tube A has a higher temperature and more energy would be available in the enzyme molecules at greater temperatures, thus increasing enzyme activity. Remember, enzymes is molecules too. You can't have too much though, the enzyme would begin to denature. Let me look at part 3 here. So state two roles that the moisture from the cotton wool plays in the process of germination. Well, water is absorbed by the seed or the tester to allow it to split. And water is also used during enzyme activity to break down or hydrolyze, that means to break down using water, large complex molecules in the food storage, that's like starch for example, into smaller simpler molecules, let me say like glucose for example. Now next one here, remember water is used as a solvent and a transport medium, so it'd be able to transport those smaller simpler molecules around the seed as well. Let's take a look at B. The corn seedlings which germinated are placed in a pot and exposed to unidirectional light as shown in figure 2. Basically that means light coming from one direction. So here we have the light bulb, we have the corn shoot and a desk lamp. And we could see clearly that the corn shoot's growing towards the opening where the light is entering, in the direction of light basically. So what type of movement is shown here? Well, this is a growth movement or a part movement, and this is phototropism. It's just one mark. Answer that, you're good to go. State a hypothesis that the students could test by using the apparatus and material shown in figure 2. Now remember, a hypothesis is just a statement that should be about something that's testable, observable, that you're eventually able to draw a conclusion from. In the case here, a hypothesis could be simple as plants grow towards the direction of a light source. Part 3. The students want the seedlings to grow towards the ceiling. Suggest how they can manipulate the apparatus to get this result. Well, basically, you don't want to have a unidirectional light source coming from the left or the right. You could put the light source above it and it will grow, of course, towards the light above it. Or you can simply cover the opening of the box and it will just grow upwards because no auxins will develop on either side of the meristem of the plant. Part 4. The seedlings shown in figure 2 use all the nutrients stored in the cotyledons. Discuss the process by which these plants can make more nutrients. Well, let's talk about photosynthesis here. And remember, photosynthesis has two stages. It has a light dependent and a light independent stage. So during the light dependent stage, chlorophyll, which is the green pigment in the chloroplast, absorb light from the sun. That light is used to split water molecules into oxygen and hydrogen, and that is called photolysis. So remember, water is H2O, so that gets split up into the hydrogen and the oxygen. The oxygen diffuses out of the stomata, that's actually the byproduct of photosynthesis, but hydrogen remains in there, and that would eventually combine with carbon dioxide with the use of enzymes such as Rubisco. We will combine with the carbon dioxide taken into the stomata. So now you'll have a compound with carbon, oxygen, and hydrogen, and that would be a carbohydrate, in this case a glucose. So this would be the light independent stage where these enzymes work to combine the hydrogen and the carbon dioxide. And eventually that glucose, which is a monomer, which means like it's a single molecule, is not linked to anything else, would eventually, a bunch of them would link up to form starch, which is the storage molecule for sugars. So part 5, discuss the processes by which the raw materials required for making nutrients reach the leaves of the plant. So water is absorbed by root hair cells in the roots via osmosis. Water then enters the xylem vessels from the roots, where it is moved to the leaves via transpiration of pull and capillary action and root pressure. So remember, the two raw materials needed is water and carbon dioxide. So carbon dioxide diffuses through the stomata, and that would be through the lower epidermis of the leaf, and is then moved through the spongy mesophyll, the intracellular spaces, there because a lot of air spaces, and up to the chloroplast and the palisade mesophyll. The palisade mesophyll is densely packed with chloroplast, so that's where the party happening, that's where most of the photosynthesis is occurring. Alright, see part 1. Starch is the most common carbohydrate that is found in the leaves of green plants. Describe a test that the students could do in the laboratory to investigate whether starch is present in the leaves of the seedlings. This is very common. You would have done this in school already, but 1, you're going to boil the leaves and that weakens the cellulose in the cell walls. 2, you're going to boil the leaves in ethanol. This is going to remove the chlorophyll, and that's going to give the leaf a more pale green or paler appearance. This is so you can see the color changes more easily. 3, we're going to place the leaves on a white tile and using a dropper apply a few drops of iodine solution. Iodine solution has a kind of yellow-orange color, so when the presence of starch is detected, that yellow-orange color is going to turn blue-black, and that's going to indicate a positive presence of starch. And let's look at part 2 and 3. Name one plant organ other than leaves where starch may be found. This is going to be in plenty parts of the plant. So seeds, fruits, roots, stems, rhizomes, tubers, anything like that, any storage organ would contain starch. And part 3, instead of starch, animals store complex carbohydrates such as glycogen. Name 2 organs in an animal's body which store glycogen. This would be the liver, which stores the majority of glycogen in the body, and any muscle tissue really. So for example, muscle tissue in the heart. I know muscle tissue is not an organ, but basically any organ that contains some kind of muscle tissue would work here. And this is question 1 for 25 marks. Let me look at Jan 2015. This is question 1D. Figure 2 shows the growth of a seedling over 8 days. Here we can see the growth of the radicle, which forms the rudimentary root. Here we have the seed coat, of course. We have the cotyledons coming out here. These act as food storages and very rudimentary leaves. And then we have the true leaves or the primary leaves being formed. Of course, the stem is called a plumule. So the question here asks to measure the length of the radicle of the seedling on each day and record your measurements in table 1. Of course, you wouldn't be able to do this on your phone or your computer, but if you get your hands on a physical paper, what you're doing here is that you're measuring from the beginning of the root here to the very tip here. And then we're going to fill out the table. So this is what the table is going to look like. The length of the radicle in centimeters. You just fill out this table and you get an extravagant 1 mark for using a ruler. So the next part here is to plot the data in table 1 on the graph paper below. Put days on the horizontal x-axis and of course the length of the radicle on the y. Remember, there's the responding variable, there's the manipulated variable. In the case here, you only have 1 set of data, so you don't need a key or legend. You just plot the points and you're good. For your title, make sure that make sure you have an appropriate scale. Make sure that you occupy at least half of each axis when you're drawing it. Don't draw the thing big, that you can't fit the whole thing. Don't draw the thing chunky where you can barely see it. Make sure you occupy at least half of the axis. And this here would be a line graph. You use your ruler, you connect the points and you're good to go. State 2 factors other than water that are required for the germination of the seed, well, oxygen and a reasonable or suitable temperature. So the question here is to describe an investigation to test the hypothesis water is needed for the germination of seeds. You're going to obtain 2 samples of loam soil of similar, well, there should be similar mass and, particulate size, and heat 1 sample until all of the water is evaporated. So basically you're going to have a regular sample and you're going to have a desiccated sample, a dry sample. So plant 20 healthy seeds in the dry loam soil and 20 healthy seeds in the regular one. Water the seeds in the regular loam soil every day, but not the seeds in the dry soil. And then they could observe the rate of germination or how many seeds germinate over a 2-week period of time. And they could record how many, every day or every 2 days or so. And part E suggests why the cotyledons or the seed leaves become smaller as the primary leaves increase in size. Well, this is because cotyledons are temporary food storages that are broken down by enzymes before the primary leaves are formed. When the primary leaves are developed, photosynthesis would occur and nutrients can be synthesized for the plant. So cotyledons, you could think of those as training wheels, and eventually you throw off them training wheels when you learn how to ride the bike. They're just there for to help you in the beginning to get a little energy. You need them again because now you could do the thing for yourself. Throw them away and now you could photosynthesize. And there you go. Alright y'all, so here we have January 2016 and this is our next growth question here. So let's look at the data. So the data provided in table 1 were collected to investigate the growth of plants in light and dark conditions and to make comparisons with the growth patterns in humans. So here we could see the days of growth from, it goes in 5s, 5, 10 until 30. We have the height of the plant grown in sunlight and in darkness, and then we have the length of a human baby. So if we just looking at the numbers here, we just observe what's going on. We could see that the height of the plant grown in sunlight, we could see that there's a slow but steady increase that happens basically the whole 30 days. As for the height of the plant grown in darkness, we could see that there's a greater increase in the beginning because here's 10 to 12, a gain of 2 centimeters. Here we have a gain of 3. And then here we have a gain of 3 centimeters. Here we have a gain of 4. So there's a more rapid increase in the beginning for the plant grown in darkness. So that's interesting, but we'll explain that just now. But at this point here, it plateaus and then it decreases. The height actually decreases, whereas in the sunlight it increases. The length of the human baby, of course, increases steadily throughout. So the question here is, on the grid provided below, plot the data obtained for both plants on the same graph using appropriate scales and legends. So a graph question, this goes for 5 marks, and let me just zoom out a little bit so you can see the graph. And here we have the length of the plant in centimeters, and here we have the time in days. You want to make sure and have your responding variable on the y-axis and your manipulated variable, in this case here time, on the x-axis. You'll notice here that the blue line represents the plant grown in darkness. You see it never really attains the height of the plant grown in the sunlight. It plateaus and then the height decreases. Whereas for the plant grown in sunlight, it increases not as rapidly as the one grown in darkness, but then it steadily increases at a certain point. We'll see that there's a scale and a key. Whenever there's 2 data sets, you want to ensure that you have a key. So the 5 marks here really is making sure that you have an accurate scale, making sure that the points are plotted accurately, making sure that you have the axes oriented accurately, responding variable here, manipulated here. You have the units and the labels for them, and that you have a key or a legend stated here. You must indicate which one is which. In the case, you can use a dotted line and a solid line for one, or you can use different types of plot points. You could use X's for one, or dots with circles for the next one. It is also a very wise idea to put a title, and the title in the case here will be a descriptive title, in the case here, graph of the growth of plants under varying conditions over an extended period of time. So usually whenever you draw a graph, you'll have to indicate any patterns or trends on that graph. So let's go through that here now. So describe the pattern of growth obtained for each plant. So in the light, growth increased slowly during days 5 to 15 to a height of 15 centimeters, and then there was a steep increase during the days 15 to 30 until a maximum height of 30 centimeters. So usually when you're indicating trends, it's good to indicate at what points those increases or decreases occurred, or if it plateaued. In the dark, during days 5 to 15, growth rate was slightly higher until a height of 17 centimeters was attained. The height plateaued, that means it didn't increase or decrease, at 17 centimeters until day 20, and then it decreased to 15 centimeters by day 30. Now, even though this is just 2 marks, this might be a little too much information for 2 marks, but you all will know how much to put in case you get like this for 3 or 4 marks. You need to have some detail and some figures in there when you're describing patterns or trends. So the question here is now to explain the differences observed in the growth of the plants under these 2 conditions. So, well, the one with the bright light is easy because photosynthesis would have been occurring in the presence of light. This would have allowed the conversion of light energy into chemical energy, in this case here, sugars, glucose, starch. The plant would have more energy, of course, for the enzymes to work at their optimum rate, and they could synthesize more proteins for growth as a result. In the dark, however, photosynthetic rates would be very low. There wouldn't be any light energy for the chlorophyll to capture, for photolysis and the splitting of water and all of that. So stored energy resources are utilized to obtain energy from the growth. So you'd have energy stored in the leaves, you'd have energy stored in the seed, you'd have energy stored in the stems, and so on. So they'll be using that up, kind of like how the humans use up the fatty tissue when they're starving. But at a point, these resources are going to be expended and no more growth is going to occur. It's going to cease. So the height is going to start to decrease, as it starts to wilt, as leaves and stems lose mass and start to shed. So as a result, that's what caused the drop in height in the graph. So let's look at questions C and D. Complete the following definition for growth by writing the correct words from the listing brackets. So growth may be defined as a permanent increase in the mass and volume of an organism when food is absorbed and converted to living matter. Transient, by the way, means like temporary or non-permanent in the case. Suggest why the measurements of length taken for the plants are incomplete, unlike the measurements taken for the baby. Well, this is because they would have measured the entire length of the baby, but they only really measure the stem of the plant. The plant is more than the stem because the plant is also the roots. In this case here, they didn't measure the roots. E suggests 2 ways in which this experiment could be refined to obtain more accurate data for measuring growth. Well, they can simply measure the roots as well. That might be a little difficult because then they would have to, dig into the soil and disrupt the actual growth of the plant as a result. So we could also measure the growth of the plant at smaller time intervals. Remember, they measured every 5 days here. Well, you could probably measure every day instead of every 5 days. They could also take other factors into account, such as the dry weight of the samples, to observe the increase in biomass. Alright, so let's look at F. Describe one way in which the pattern of growth in plants differs from the pattern of growth in animals. Well, animal growth occurs at all regions of animal tissue. So the animal expands in all regions. But the plants, however, growth really only occurs on the meristematic tissues, the apical meristems. Those are on the tips of roots and shoots. They don't occur across all regions like an animal does. Alright, so let's look at G. Describe an experiment to demonstrate the response of plants exposed to light on one side only, where you're going to obtain 2 similarly grown plants of the same species and of the same height. You're going to enclose one plant in a box with an opening exposed to a light source, kind of like the previous question. You're going to enclose the other in a box with either no opening or an opening covered with black cloth or on opaque cloth so that no sunlight can go through. And you're going to record the direction of the growth of the plant, each plant over time, maybe each day or every few days. And how does this response to light benefit plants in a forest? Well, when plants grow towards sunlight, it maximizes light absorption by chlorophyll for photosynthesis. Let's look at part 3. Explain how and why the response of invertebrates like earthworms to light differs from that shown by plants, where we know that earthworms and invertebrates have a negative response to light. They move away from it. So invertebrates undergo locomotory responses away from light. Locomotory refers to like locomotion, which means the movement of the entire body, where they can move from one place to another. Remember, plants can't do that. They only can move parts of their body. So plants undergo positive growth responses to light. The invertebrates will be more visible to predators in bright conditions. This is why they move away from the light. It can be more easily spotted and would be more prone to desiccation or drying out. Also, where there's bright light, there's likely to be higher temperatures. So they more move towards, moist, dark areas. State one role that light plays in human growth and development. Either one of these would do: the synthesis of vitamin D by the skin, or melanin. As you go out in the sun, your skin produces more melanin, and that's actually how you get a tan. And that is 25 marks. Alright, so let's look at some questions here dealing with photosynthesis. Well, 5A, this is June 2016. With the aid of a chemical equation, explain what is meant by the term photosynthesis and discuss how a die-cut leaf is adapted to capture maximum sunlight and carbon dioxide for the process. Basically, listen the adaptations of the leaf. So the definition of photosynthesis: the conversion of light energy to chemical energy by green plants and also some photosynthetic bacteria, is basically the conversion of carbon dioxide and water to sugars using sunlight. The equation here, now, these numbers here at the end, like this CO2, well, you know this already, but these are supposed to be subscript, right? So CO2 and a small 2 underneath. So 6 CO2 plus 6 H2O gives you C6H12O6 plus 6 O2, and this is basically 6 molecules of carbon dioxide plus 6 molecules of water gives you glucose plus 6 molecules of oxygen. So the adaptations of the leaf. And let's look at the amount of marks here, right? So this is 11 marks. So we can say that the definition is 1 mark, the equation is probably 2 marks, and that leaves us with 8 marks left. That means we want to have at least 4 adaptations listed and explained very well. 2 of them have to relate to sunlight, 2 of them have to relate to carbon dioxide. So first, let's start with the ones with sunlight. So I have 3 here. The leaf is oriented to the sunlight by the petiole to maximize exposure for light absorption. Basically, the leaf undergoes like slightly phototropic movements to bend the leaf towards the light. The upper epidermal cells of the arm of the leaf contain no chlorophyll, and they are transparent cells. And these basically allow light to penetrate and reach the palisade mesophyll cells. Imagine like you have a nice glass ceiling above. And the leaf has a broad lamina, so the leaf itself is broad, to provide a large surface area for light absorption. The next ones we can see here relate to carbon dioxide. The leaf has numerous stomata in the lower epidermis to provide a large surface area for carbon dioxide diffusion. And the leaf has numerous air spaces or intracellular spaces in the spongy mesophyll layer, which provide unobstructed pathways for the diffusion of carbon dioxide and also water vapor. Any 2 of these up here and any 2 of these well explained should give you all your marks. And let's look at B. Outline how decomposers assist in the cycling of carbon dioxide to make it available for green plants. So decomposers break down dead organic matter of organisms. These dead tissues contain carbon, and they use those tissues as a substrate and they digest them. And this wouldn't just be dead carcasses or dead bodies, it would also be like shed material, like leaves, bark, skin, excretory material, digested material like feces. So they use these materials for respiration as they break these organic materials down into simple inorganic material, one of those being carbon dioxide. Carbon dioxide is released during this process where it is released into the atmosphere, and eventually it will be absorbed by the leaves of green plants during the process of photosynthesis through the stomata. If you have all of this well explained, I'll give you 4 marks. Alright, here we have another photosynthesis question. This is June 2015 question 2. The following chemical equation for photosynthesis represents the reactants used and the products formed. This is the same equation that we would have just written. So name two conditions which are not shown in the equation that are essential for this process. That will be light energy, presence of chlorophyll, or suitable temperature range. Any two of those would do. Name the gas given off in this process and state which of the reactants produce this gas. Of course, the gas is oxygen, and remember that oxygen is formed from the photolysis of water, where that water is split by sunlight, split using sunlight into oxygen and hydrogen. Let's look at 3 and 4. Explain two ways in which plants utilize the glucose produced during this process. Well, glucose is used for respiration. It's used to produce ATP during the process of respiration. And glucose is converted into starch for energy storage, where it's also converted into cellulose to build cell walls. Any two of those well explained should give you a marks. And part 4, state 3 environmental factors which affect photosynthesis. We have how well the plants are exposed to the sunlight, how well the leaves are exposed to the sunlight, the presence of carbon dioxide in the atmosphere, and whether or not the atmosphere has a suitable temperature range for optimum enzyme activity. Alright, so here we have question 4 of January 2017. It's not so much a photosynthesis question, but it does deal with it, and it also does deal with blood. So might as well just throw it in here, you never know. But 4a, describe how oxygen in the atmosphere becomes available to human blood for delivery to the cells. Explain how this process differs from the way plant cells obtain oxygen for respiration. Well, first of all, we know that humans, mammals, we get oxygen from the air, we breathe it in. So oxygen is inhaled, through our nostrils, it goes down our trachea, our bronchi, bronchioles, eventually it ends up in our lungs, alveoli, or air sacs. From there, the oxygen diffuses through the moist alveoli linings and into the capillaries. So remember, blood is flowing away from the heart at this point, and that will be deoxygenated blood. And red blood cells would be flowing through single file in those capillaries. Now what would happen now is that the deoxygenated blood is pumped away from the pulmonary arteries from the heart to the alveoli. And those red blood cells in the capillaries contain the protein hemoglobin. So hemoglobin in the red blood cells is able to bind with oxygen. That hemoglobin there, that would be deoxygenated at this point, right? So they would have what you call a high affinity for oxygen. They would take it up very easily. So oxygenated blood is then returned to the heart via the pulmonary veins, and then is pumped out of the heart into the body, towards the body cells via the aorta, that's the main artery. So oxygen then dissociates from the red blood cells, basically that means like it unbinds from the red blood cells, and it diffuses into the body cells through the surrounding tissue fluid. So remember, the capillary network have like very, very small tubes. I like to think of them as like little small roads leading to cells. Each cell is like a house, and each capillary is like a small little road. And there's a little gap between the road and the house, just like there's a little gap between the capillary and the body cell. So that gap there is actually filled with tissue fluid. So whatever materials, whether it's glucose or gases, must diffuse through that tissue fluid to reach to the cells or the capillaries, or vice versa. So in plants, however, of course, we don't have all these networks. So in plants, oxygen diffuses through the stomata on the lower epidermis of the leaves, just like carbon dioxide. The oxygen moves through the intracellular air spaces between the spongy mesophyll cells. And this is because of, diffusion, because diffusion is the transport of a fluid from a higher to a lower concentration. So oxygen travels from higher concentrations in the atmosphere to lower concentrations in the leaves. Here we have January 2016, question 3. We have a question that mixes cells and photosynthesis. So here we have a diagram of a guard cell seen on a dicot leaf, taken from a plant that has been watered 12 hours before and placed in sunny conditions. The first question is to identify the structures labeled 1, 2, 3, 4, and 5 in figure 2. Write your answers in the spaces provided. In my opinion, this is a very bad diagram, a very poor diagram. But you know what else is bad? when you don't know how to write, you know how to spell, you know how to do nothing. So you know what you have to do? Sign up for a free trial of Grammarly. Yes, this channel is sponsored by Grammarly. Yes, you sign up for that. You see all who gain 4s and 5s in English? Nah man, sign up for Grammarly right now. That's the only way you're going to get employment. Alright, all you back to the review. Well, here we have the 5 things, and 1 is the cellulose cell wall, 2 is the cytoplasm in the cell, 3 is the chloroplast, 4 is the stoma, that's the singular for stomata. Remember, these are the guard cells neighboring it, like, you know, it's sandwiched in the stoma basically. And 5 is the nucleus. A very, very bad diagram. So the next question here is state one function of each of the structures labeled 1 and 4. Well, 1 is the cellulose cell wall, and that prevents the cell from bursting. It basically resists any internal water pressure. As you know, when water flows into the cell, the cytoplasm expands and the cell is liable to burst. This cell wall prevents that from happening. And 4 is the stoma, and that allows the diffusion of gaseous materials, for example, carbon dioxide and water out of the leaf. It also allows some intake of oxygen and also lets out oxygen during photosynthesis. Anything like that would work. It's just 1 mark each. So the question here is, in the space below, draw an annotated diagram to show the appearance of the guard cells after the plant is watered in the early morning. Use an arrow to show the movement of the water in the cell. So the water is going to move, this is the arrow here, the water is going to move through the cellulose cell wall, and remember this is freely permeable, and then it's going to move through the partially permeable cell membrane, right, of the guard cells. So here we have the cell wall, and remember the cell wall is going to prevent it from bursting. The cell membrane is no longer visible, it's pressed against the cell wall as a result because the cytoplasm is expanding, making the cell very turgid. The stomata is much more open now, so this orifice here is going to be a lot bigger, and the curved wall of the guard cell is going to be closest to the stoma, as you see in here. Cytoplasm has expanded as water moved into the guard cell by osmosis. Explain why the behavior of an animal cell would differ from that of a plant guard cell when they are placed in dilute solutions. Dilute basically means there's a lot of free water molecules, has a high water potential. Well, in both cases, water is going to enter the cytoplasm of the cell from the distilled water solution, a dilute solution. The cellulose cell wall of a plant cell resists internal water pressure as the cytoplasm expands, but since animal cells have no cell walls, they are liable to burst if the pressure is too high. This is called lysis. Alright, so we're just going to look at a few parts in June 2021. Although it's less likely for them to repeat questions just a year before, but you never know. It's CSEC, you never know. Alright, so this is question 1C of July 2021. The leaf shown in figure 1 on page 4, well, this is the leaf here, just a normal leaf. And I wanted it to label it. We have the apex, the lamina, the midrib, the petiole. So they wanted it to suggest one way in which the leaf is adapted for each of the following functions. So you have gaseous exchange, and the adaptations would be numerous stomata on the lower epidermis, numerous intracellular air spaces in the spongy mesophyll, and to prevent excess water loss, well, above the upper epidermis we have a waxy cuticle that is usually waterproof, and then we have the presence of numerous guard cells on the lower epidermis, and remember those could close, right, kind of like gates closing to prevent excess water from leaving the plant via transpirational pull. Now let's look at D. Green plants produce food by photosynthesis suggests 3 reasons why there's still a need for them to store food. Well, they have to provide nutrition for embryos or seeds. For trees or plants that live in temperate regions, they would also have periods of scarcity like in winter, and they have to store a lot of food then. You also have plants that are susceptible to drought. It's beneficial for them to store a lot of food during those times. Kind of like bears when they undergo hibernation. And then there's periods without sunlight or when no photosynthesis can occur. So during the night, for example, especially in countries where the night period will be very, very long, they have to be able to store a lot of food in their stems and in their bark and so on as a result. So let's look at E. Suggest 3 reasons why the initial carbohydrate product of photosynthesis, which is glucose, is not stored in green leaves. Remember, say starch is stored, so why not glucose? Well, this is because, well, glucose is a very reactive molecule, right? It is soluble and is easily dissolved in water and easily transported around, but it's very reactive. And what happens here is that because they are so small and much smaller than starch, they can easily exit the cell membranes as a result. And remember, the cell membranes are partially permeable. They want molecules that could be stored as large starch grains that would not be able to leave through the gaps in the cell membrane. So also stored glucose disrupts osmotic potential and will cause excess water to flow into the plant cells. Basically, you'll have a very low water potential inside the cells, excess solute basically, and that would cause more and more water to flow in. Right, so you don't want that to happen. So these 3 explained very basically should give you 3 marks. Let's look at some questions that deals with cells and deals with osmosis basically. There are many questions that deal with this, but I've chosen 2 that, you know, pretty much cover the bases here. So this first one is from June 2018, question 2. Figure 3 shows 3 different types of cells, A, B, and C. Name the types of cells labeled A, B, and C. So A is an animal cell, B is a plant cell, C is a bacterial cell. The reason you could tell A from B, by the way, is B has the cellulose cell wall around it. A doesn't have a cell wall. The thing is, C also has a cell wall, the bacterial cell, but it's made up of different materials, not cellulose. You could also see the presence of chloroplast in the plant cell here in B. So let's look at 2 and 3. Name 2 structures that are common to the 3 types of cell. Well, that will be cytoplasm, a plasma membrane or a cell membrane, and ribosomes for synthesizing protein. Although the ribosomes in the bacterial cell are a lot smaller, but you know, they still contain ribosomes. So any 2 of these would work. State 2 differences between the type of cell labeled A, that is your animal cell, and the type of cell labeled C, that is your bacterial cells. Well, animal cells contain a nucleus. Well, first of all, animal cells are eukaryotic cells, right? That means that they are a lot larger. They have organelles, double membrane organelles, such as Alright, state 2 differences between the type of cell labeled A, that is an animal cell, and the type of cell labeled C, which is a bacterial cell. Well, first of all, an animal cell is eukaryotic and a bacterial cell is prokaryotic, right? And one of the main differences between that is that animal cells have a true nucleus and they have membrane-bound organelles, double membrane-bound organelles. And bacterial cells do not have a true nucleus, right? Bacterial cells, on the other hand, have their DNA in a nucleoid region, basically in little circular plasmids or linear string-like DNA kind of deposited amongst the cytoplasm in the middle. And eukaryotic cells tend to be larger and much more complex than prokaryotic cells. So animal cells contain a nucleus and double membrane-bound organelles such as mitochondria, but bacterial cells do not contain either. They have DNA in a nucleoid region. Talking about bacterial cells here. Bacterial cells, on the other hand, well, they have a cell wall, while animal cells do not, right? 2 basic differences, and you're good. Alright, so B, tissues made up of type B cells as plant cells are placed in a container of concentrated salt solution and left for 1 hour. Remember, concentrated solution would have very, very few free water molecules. So this is said to have a low water potential. Describe 3 changes in the appearance of the cells after 1 hour in the concentrated salt solution and give an explanation to account for these changes. Well, in the concentrated salt solution, water would flow out of the plant cell and into the solution, basically from a higher to a lower water potential due to osmosis. Right? So water flows from a higher to a lower water potential through the plant cells partially permeable membrane. So as a result, the three changes: the cell is going to undergo plasmolysis. That means it's going to shrink, it's going to shrivel a little bit. The cell membrane is going to retract from the cell wall. The vacuoles are going to lose water and going to become smaller as water flows out of them. And the cell is going to lose cytoplasm or cytosol, and the tissues will become flaccid as a result. So you could say that the cytoplasm is going to shrink as a result. Tissues made up of type A cells as animal cells and type B cells as plant cells are placed in a container of distilled water and left for 1 hour. Describe how the appearance of the animal cells is expected to differ from the plant cells. We did something like this just now. But in this case, you would explain, well, if they place in distilled water, that would have many free water molecules, so this would be a high water potential. So in both cells, water will flow from the distilled solution, higher water potential, and into the cells, which have a lower water potential due to osmosis. And both cells are going to gain mass because they are absorbing water as a result and begin to swell. Hydrostatic pressure within the cell is going to increase. However, the animal cell, which is A, will likely burst, while the plant cell B would likely not. Remember, that's because the animal cell doesn't have a cellulose cell wall, while the plant cell does have a cellulose cell wall that is able to withstand the turgor pressure. Alright, let's look at, this will be Jan 2017, question 2. State 3 differences between a plant cell and an animal cell. Basic question. Plant cells contain chloroplasts and cellulose cell walls, while animal cells do not possess either of these. These would actually count as 2 differences right here. Plant cells have a large permanent vacuole, while animal cells have small temporary vacuoles. The plant vacuole, by the way, is also like central to the cell, but the animal cells vacuoles are distributed through the cytoplasm, not central. And plant cells contain starch grains as their energy reserves. Animal cells contain glycogen granules as their energy reserves. Part 2. State 2 differences between a prokaryotic and a eukaryotic cell. Eukaryotic cells contain a true nucleus with DNA enclosed in a nuclear envelope, both DNA and RNA. They also contain double membrane-bound organelles such as mitochondria and chloroplasts. Prokaryotic cells' DNA, on the other hand, is not found in a nucleus but is distributed in a nucleoid region as plasmids or circular DNA or stringy linear DNA, whereas they do not have membrane-bound organelles, so they don't have mitochondria and chloroplasts and things like that. State the function of each of the following. The cell wall prevents the cell from bursting by resisting inward hydrostatic or water pressure. The cell membrane regulates the entry and exit of materials in and out of the cell, basically allows small non-polar molecules enter and exit, but prevents large polar molecules from entering and exiting. Alright, so C here basically, accounts for the same thing that we explained just now, so I'm not going to repeat it. But you could pause the video and read the responses. Alright, so let's get into these carbon cycle and climate change and conservation questions here. So we are looking at June 2019. This is question 2. Figure 3 is a diagram of the carbon cycle. And here we have 4 parts labeled here. 1, 2, 3, 4 that we have to label. But just to go through it here basically, let's start with atmospheric carbon dioxide. Well, this is going to get absorbed by the leaves of green plants, and of course one therefore is going to be photosynthesis. And these green plants are going to be eaten by animals, going to pass on carbon through the food chain, through the process of consumption. Of course, the plants and the animals involved here have to respire, and this would be respiration, and that would release carbon dioxide back into the atmosphere. When these animals and plants die, they undergo decomposition, and bacteria and fungi break down the organic material into inorganic substances and release carbon dioxide back into the atmosphere. So 4 is going to be decomposition. Eventually, this decomposed material is going to fossilize, undergo fossilization, is going to get pumped up as fossil fuels or oil, and that's going to be burnt, which is tree, and this is going to be combustion. They're going to get burnt, and that's going to release the carbon compounds, carbon dioxide, back into the atmosphere. So just to go through each one here. 1 is going to be photosynthesis, 2 is going to be respiration, 3 is going to be combustion, and 4 is going to be decomposition. Name one type of organism responsible for decomposing organic waste. Bacteria, fungi, either one of those would work. Explain 2 ways in which the type of nutrition carried out by the organism named in B1 is different from that carried out by plants. Well, we have 3 types of nutrition we could talk about. We have autotrophic nutrition, heterotrophic nutrition, and saprophytic nutrition. Autotrophic means self-feeding. Heterotrophic means feeding from another. Saprophytic is kind of like heterotrophic, which means that it basically breaks down dead matter. It uses that as a substrate or a digestive material. So how is it different? Well, plants use autotrophic nutrition, and this is basically photosynthesis. It converts inorganic raw materials such as carbon dioxide and water into organic materials such as sugars, in the case of glucose and starch, using sunlight. They do not depend on other organisms for their nutrition. They basically convert raw materials into organic products. Saprophytic nutrition basically decomposes, converts organic matter in dead tissue and basically any shed material like leaves and bark and feces and things like that into inorganic materials such as carbon dioxide. It does not require sunlight to do so, like photosynthesis, and must rely on other organisms for their nutrition, so they're not self-feeding as a result. So there we have 2 ways in which they are different. You have that well explained as 4 marks. Part 3, state 2 reasons why the type of organism named in B1 is important in an ecosystem. Talk about decomposers. So decomposers prevent the accumulation of dead matter. Basically, it prevents too much dead material from piling up. It also helps to cycle carbon back into the atmosphere as carbon dioxide. That's the 2 reasons there. And C, part 1, suggests 2 reasons why carbon dioxide is described as a greenhouse gas. So as you should know, a greenhouse traps heat, and how it does that is because short wave rays from the sun can penetrate the greenhouse glass, but when it gets reflected, it turns into long wave sunlight. Basically, this is like infrared and ultraviolet and things like that, and that cannot penetrate. Carbon dioxide acts like that greenhouse glass, and it turns the earth into, you can think of it as a greenhouse. Basically, it traps that sunlight. The sunlight can enter but it cannot exit. Think of it like a parked car in the hot sun. The heat could come in but it can't come out, causing the car to heat up. That's a good example. So carbon dioxide, along with other gases, form a greenhouse layer in the atmosphere. It prevents long wavelength sun radiation from escaping the atmosphere. This contributes to a rise in temperature and climate change as a result. With reference to the carbon cycle, suggests 2 strategies individuals can use to reduce the amount of carbon dioxide in the atmosphere. Well, they can reduce fossil fuel usage, they can carpool, use public transport, walk, use bikes. They can use electric cars, hybrid cars, anything that basically reduces fossil fuel usage. They can consume less electricity by using more energy efficient appliances, energy efficient cars, switching from incandescent bulbs to fluorescent and LED light bulbs. They can turn off the air conditioning units when not in use, things like that, right? Basic things. And let's look at Jan 2016, question 2. Same topic. The diagram shown in figure 1 represents part of the carbon cycle. Complete the diagram by writing the appropriate term in each of the boxes labeled A, B, and C. Kind of the same thing that we did before. So A is going to be photosynthesis, because this is going to be the process that takes carbon dioxide out of the atmosphere and puts them into green plants. B is going to be respiration, in which animals and plants are going to do to put carbon dioxide back into the atmosphere. And C represents the burning of the fossil fuels, and that's going to be combustion. Write a chemical and a word equation to summarize each of the following processes. Photosynthesis, did this already. Carbon dioxide plus water. With the help of sunlight and chlorophyll gives you glucose plus oxygen. Here we have the chemical formula. And aerobic respiration is kind of the reverse of that, where we have glucose plus oxygen gives you carbon dioxide and water. C6H12O6, that's glucose, plus 6 molecules of oxygen gives you 6 molecules of carbon dioxide and 6 molecules of water. C. Outline 2 outcomes of the removal of trees by cutting and burning. Well, first of all, burning the trees is going to release carbon dioxide into the atmosphere. That's going to contribute to the greenhouse effect because you're going to get a huge greenhouse layer of carbon dioxide in the atmosphere. That's going to trap long wave radiation, sunlight, and is going to trap heat, and that's going to lead to increase in temperature and climate change. Now, cutting down the trees is going to reduce the number of trees that would be available to remove carbon dioxide from the atmosphere. Remember, trees undergo photosynthesis. You remove them, you're going to have less trees photosynthesizing, and thus a lower rate of carbon dioxide being removed from the atmosphere. D. Suggests 2 ways in which small island states, such as those in the Caribbean, may be affected by large-scale climate change. But climate change leads to increased temperatures in both the ocean and atmosphere. This leads to rising sea levels, which contributes to flooding and the destruction of coastal ecosystems. You also have haphazard weather patterns. You have an increase in hurricanes, for example. Of course, all of that could lead to devastation of the economy of small island states, not just lives lost. Next, we have ocean acidification. Basically, when carbon dioxide gets absorbed into oceans, this lowers the pH of the water. This results in coral bleaching, and as a result this would impact tourism revenue. And let's look at E. Outline 2 ways in which the improper disposal of household waste contributes to land pollution or water pollution. This could be basically littering, or it could be runoff of agricultural products or runoff of detergents or anything like that, basically dumping all these things into the water. Well, non-biodegradable refuse or waste may clog waterways and contribute to flooding because they take a very long time to break down. This would be a plastic bottles, a glass bottles, and so on. Agricultural chemicals containing nitrates and phosphates may run off into ponds and contribute to eutrophication. This is algal blooms. This could also be due to the runoff of sewage, the improper disposal of sewage. But basically what happens here is that the algae overgrows and uses up the oxygen in the water, causing the other organisms to asphyxiate, and you wouldn't get anything else really surviving in there. Eventually what's going to happen is going to get a buildup of bacteria that's going to use up even more oxygen in the water. Suggests 2 ways by which people could dispose of their household waste in an environmentally friendly manner. You can reuse these materials at home. They can refill plastic water bottles, use them as plant pots, basically repurpose them, find a next use for them. Or they can compost biodegradable materials instead of tossing them out, or they can send them to a landfill to be buried, or they can send them to a recycling center such as plastic products or glass products. Any 2 of these would work. (6a) State 2 causes of anemia and describe 2 signs or symptoms of the disease. So we have iron deficiency anemia and we have sickle cell anemia. Iron deficiency anemia is just the lack of iron in the diet. Sickle cell anemia is due to the inheritance of 2 recessive sickle cell alleles. One of them is a nutritional deficiency disease and condition, and the next one is a hereditary condition. The signs and symptoms of each, however, are somewhat the same. Basically, you have a low concentration of hemoglobin molecules, or you have abnormal hemoglobin molecules. You have a lower red blood cell count, that should be count here, and you have fatigue or lower respiratory rates. There should be like lower normal red blood cell count because you'll have sickle cells being replacing the normal red blood cell for sickle cell anemia. So sickle cell anemia is caused by the presence of a recessive allele. Persons who are heterozygous and carry the sickle cell allele are more resistant to a type of severe malaria than persons who are homozygous for the normal hemoglobin allele. Heterozygous means that they have a dominant allele and a recessive, right? And typically they are just carriers of the disease, they don't actually suffer from it. So state what is meant by each of the following terms. Well, right here, homozygous, homo means same, hetero means different. Homo means two identical alleles for a common gene. So for example, capital B, common B, for example. Heterozygous means they have different alleles for a given gene. So for example, capital B, common B, two different alleles. With the aid of a genetic diagram, show how a man and a woman who do not have sickle cell anemia can have a child who has the disease. So in the case here, we're going to have the phenotypes. They're both not going to suffer from sickle cell anemia, but they're both going to be carriers for the allele. So we have hemoglobin A, and that's going to be our normal allele. This is for your normal hemoglobin. And hemoglobin S, this is going to be a sickle cell allele. If a child inherits two hemoglobin S alleles, that child is going to suffer from sickle cell anemia. But hemoglobin A is dominant to hemoglobin S. Hemoglobin S is recessive. So both parents are going to be heterozygous. They're going to have one normal allele, hemoglobin A, and one recessive or abnormal allele, hemoglobin S. In terms of the actual Punnett Square, this is what it's going to look like. So the father is going to be hemoglobin A, hemoglobin S. Mother, same thing. And the first, so remember, a Punnett Square here doesn't mean that these two people had 4 children. It means for each child, these are the possibilities of the inherited alleles. So this first possibility is going to be hemoglobin A, this is going to be a child, a normal child, normal red blood cells, so to speak, and also not a carrier. This second child is going to inherit hemoglobin A, hemoglobin S, and this child is going to be a carrier. Carriers for sickle cell anemia are said to have sickle cell trait. So this is going to be a sickle cell trait child. Same thing with this child, inherit hemoglobin A, hemoglobin S. This last possibility is going to inherit hemoglobin S, and this is going to be the child that suffers from sickle cell anemia. So the genotypes are seen here. The phenotypes, you're going to get normal. You could also see this as normal, sickle cell trait, and sickle cell. So either one is fine, but basically sickle cell trait children, they don't suffer from sickle cell anemia, so you can say normal as well. So the phenotypic ratio is going to be 3 normal to 1 sickled. That's going to be this child here, basically a 25% chance. Explain why the sickle cell allele is more common in the population in countries where the severe form of malaria is present. Okay, so the sickle cell allele, those who have sickle cell trait, right, basically they are heterozygous. What happens there is that their red blood cells are different from those that just have regular hemoglobin. Their red blood cells are a little more porous, and as a result, the malarian protozoan or parasite cannot actually survive in those red blood cells. So carriers of the sickle cell allele, which means those heterozygous for the trait, have red blood cells that are less likely to be inhabited or infected by the malarian protozoan. So it's also more likely that the heterozygous population will be more likely to survive malaria infection. So think of it like this. Those who are normal, which means hemoglobin A, if they contract malaria, they're likely to die, and they're likely not to have offspring. Of course, those who have sickle cell anemia, they're more likely not to reach the age to actually have children. So, not saying that, they would, but it's more likely, there's a higher likelihood that they would die compared to those who are heterozygous or those who just have normal hemoglobin. So the people who have really the highest survival rate in this case is the people who are heterozygous, because they can survive malaria and they don't have sickle cell anemia. So in that case there, once you have that explained, you should get 3 marks. Here we have January 2016. Question 5. Name 4 major components that make up human blood and explain how the oxygen transported by the blood becomes available to cells for the production of energy. Well, the components are red blood cells, white blood cells, platelets, and blood plasma. You can also say erythrocytes, leukocytes, and thrombocytes. Explanation. Well, in the lungs alveoli, Oxygen diffuses through the alveolar membranes to the surrounding capillaries. In the capillaries, oxygen binds to the hemoglobin in red blood cells to form oxyhemoglobin. The oxygenated blood is then pumped to the body cells via the pulmonary veins and then the aorta. The red blood cells flow through capillaries surrounding the body cells. Oxygen diffuses out of the red blood cells and moves into the body cells by diffusing through the capillary walls and surrounding tissue fluid. I basically just read all that because we explained this already. Same thing. You basically just explain how that oxygen moves around and how it gets into the body cells from the lungs. Alright, so here we have a kind of repeat of the question, so I'll just go through it quickly. Sickle cell anemia is an inherited condition in which the blood of affected persons cannot transport oxygen efficiently. Two symptoms explained already: fatigue due to reduced oxygen transfer because you have abnormal hemoglobin, and also you have the accumulation of those sickle cells because they can't flow as easily as the biconcave normal red blood cells. So you're gonna get edema, which is just swelling and joint pain due to the accumulation of sickle red blood cells in the extremities, like the capillaries there. And here we have the same kind of question from before. They say Fazul and Shana both share sickle cell trait. They carry the sickle cell trait, which means that they are heterozygous. What is the chance that a child born to them would suffer from sickle cell anemia? Use A for normal, S for sickle, and it's pretty much the same thing. You could pause the video just to see. Again, you can call each one here capital A normal, capital A, capital S. This is sickle cell trait. But remember, these are normal as well. So if you have the ratio as 3 to 1, that's fine. 3 normal to 1 sickle cell, that's fine. If you have it as 1 normal to 2 sickle cell trait, or 2 carriers to 1 with sickle cell anemia, that's fine too. So remember to always, when you're doing this question, state the parents' genotypes, state their phenotypes, draw the Punnett Square, put the gametes, put the offspring genotypes, the offspring phenotypes, and the ratio. And if you want to take it per percentage, so a 25% chance, in this case here 1 in 4, that the child will be born with sickle cell. And usually this will go for 5 or 6 marks. Once you have all of that, you're safe, you're good to go. Here we have January 2017 question 3. Part A, figure 1 shows components of blood. We have 3 labels here, A, B, and C. Identify the components of blood, label A, B, and C. A is a platelet, B is a white blood cell, and C is a red blood cell. You might be like, well, how we know which one is which? Well, white blood cells generally tend to be bigger than red blood cells. And looking at this here, we can see that specifically this is a lymphocyte because it has a large round nucleus. Part 2, name the component of the blood that is responsible for clotting. That will be your platelets. Part B, complete the flow diagram shown in figure 2 by filling in the missing words at positions D, E, and F. Well, clotting factors are released from platelets, and then we have plasma protein synthesized in the liver and circulating in an inactive form, and prothrombin, right, is going to convert to thrombin as a result, and this here is going to catalyze the conversion of fibrinogen, which is a soluble component in the plasma, into fibrin, which is insoluble. These would eventually form the little threads or the netting that's going to capture red blood cells to form the scab. So your answers here is going to be D, platelets, E, thrombin, and F, fibrin. Alright, so here we have July 2021, 5A. Red and white blood cells are important components of human blood. Use clearly labeled diagrams to illustrate the structure of a red blood cell and the structure of a white blood cell. So in the case here we have a red blood cell, we have a thick outer membrane, and we have a thinner part here in the middle. It kind of looks like a donut without the hole. There's a biconcave structure, and there's the cytoplasm with the hemoglobin inside. You can also label the parts that has the thick outer layer and the thinner region here. And this basically facilitates rapid diffusion of oxygen into the red blood cell and out as well, when it has to dissociate and go back into the body cells. White blood cells would have a more globular shape. Here we have the cytoplasm, the plasma membrane, and we have a large lobe nucleus. This would be for a phagocyte, by the way. Lymphocytes would have a rounder shape, less globular, and would have a very large round nucleus as a result. B. Explain how any two features of a red blood cell ensures that it is suitable for its major function in the human body. Remember, the major function of the red blood cell is to bind oxygen and to transport it from the lungs, the capillaries around the lungs, to the body cells. So how does it enable to do this? So ask for 2, but we have 4 here. So 1, it contains no large internal organelles, so it allows more room and more capacity for cytoplasm and hemoglobin. Basically, it increases its oxygen carrying capacity. 2, it has a biconcave structure. It allows it to be flexible and allows easy passage through the capillaries. 3, there's an indentation or sink in the middle that allows rapid diffusion of oxygen into the cell. And 4, it has a high concentration of hemoglobin. That's the protein required for the uptake of oxygen and the conversion to oxyhemoglobin. Let's look at these menstruation questions. Now, menstruation, the human female reproductive system, birth control, and STIs. So question 5A, this paper is June 2019. Describe the location and structure of the normal human female reproductive system. Well, the human female reproductive system is located in the lower abdomen. It consists of the vagina, the cervix, the uterus, 2 fallopian tubes, and 2 ovaries. The vagina is the orifice that allows the entry of semen. Basically, this is where you'd have entry of the penis during copulation, and this is also the exit point of the fetus during childbirth. The cervix separates the vagina from the uterus. The uterus lining, also called the endometrium, allows for implantation of a fertilized egg. Let's say that a sperm cell nucleus fuses with an egg cell nucleus and turns into a zygote. Eventually, that zygote will turn into an embryo, and that embryo has to implant on the endometrium, kind of like planting a seed in soil. So the uterus branches into 2 fallopian tubes, and each of these is lined with small hairs called cilia. And these fallopian tubes lead to 2 ovaries, which contain follicles and egg cells. B. A newly married couple wants to delay having children for a few years. Suggest one temporary contraceptive method that the couple could use to prevent pregnancy at each of the following stages. So preventing fertilization, preventing ovulation, and preventing implantation. So to prevent fertilization, we can use barrier methods such as condoms and diaphragms. These prevent the sperm cells from even meeting the egg cells. To prevent ovulation, basically preventing the release of egg cells, you could use birth control pills, and those could suppress hormones such as estrogen and progesterone. And preventing implantation, some birth control pills work as well. And you also have intrauterine devices or IUDs. You have other answers here that could work for this also, because some birth control pills help to develop like a kind of cervical mucus that prevents sperm cells from entering the uterus. You also have some IUDs that do that, so they're interchangeable. But C, the next part here wants you to explain each one. So explain how each contraceptive method suggested in B works to prevent pregnancy at each of the following stages. For preventing fertilization, condoms act as barriers which physically prevent the sperm cells from entering the female reproductive system, well, the uterus and the fallopian tubes, and making contact with egg cells. To prevent ovulation, birth control pills suppress hormones such as estrogen and progesterone, which either prevent the maturation of the Graafian follicle or prevent the release of mature egg cells. And implantation, intrauterine devices may secrete hormones that would thin the endometrium or the uterus lining, and this would prevent the fertilized egg from attaching to it, thus preventing the formation of a placenta and an umbilical cord. Alright, let's look at June 2017. This is question 3. Figure 4 illustrates the events that take place during the human menstrual cycle. And here we have the 2 graphs. Well, 1 graph and 1 matching illustration here. Here we can see different concentrations of hormones X and hormone Y. and we can see the thickness of the uterus lining or the endometrium that matches the timeline of this in terms of weeks. We also have the development of the Graafian follicle as time goes on, and eventually the formation of a corpus luteum. Now looking at it here, we can see when there's a rise in hormone X, at its peak of hormone X here, this corresponds with the release of the egg cell from the follicle. That's what's happening at Z. Z is ovulation. And right after that, we have a rise in hormone Y. So hormone X is going to be estrogen, and hormone Y is going to be progesterone. So identify hormones X and Y and the event occurring at Z. X is estrogen, Y is progesterone, Z is ovulation. What is the role of each hormone in the menstrual cycle? Well, estrogen helps to repair the uterus lining and also helps to thicken the uterus wall. Estrogen also stimulates the release of LH or luteinizing hormone, and that actually helps the Graafian follicle to rupture to release the egg cell, basically to stimulate ovulation. And progesterone maintains the thickness of the uterus lining, allowing the cells to proliferate and become vascularized. Proliferate means like to multiply, become vascularized means like to gain blood vessels, so for the blood vessels to dilate. So basically this means that if the egg gets fertilized, you want the uterus lining to remain thick so that implantation can occur. Progesterone allows that uterus lining to stay thick just in case that egg was to be fertilized. Name the organ that produces X and Y, both will be the ovary. Discuss the fate of the following structures shown in figure 4 if fertilization of the ovum occurs. Well, the corpus luteum is the remains of the Graafian follicle after ovulation has occurred. Basically, after the egg has been released, the casing around it, the follicle, decays to form a corpus luteum. That corpus luteum produces progesterone. It produces progesterone for typically a few days, but if implantation occurs, it continues to produce progesterone after a few months. The placenta produces progesterone after that. The uterus lining, what happens to that is that if it doesn't break down as a result, menstruation doesn't occur if the egg gets fertilized. So the uterus lining becomes thicker due to the prolonged progesterone release because the placenta and the corpus luteum keep producing progesterone. So this facilitates the implantation of the fertilized egg and allows formation of extra embryonic structures, so the placenta, umbilical cord, the amnion, a chorion, things like that. B. Some contraceptive pills contain hormones. Suggest how this may prevent pregnancy. Well, they may prevent ovulation from occurring, or they may thicken cervical mucus to prevent the entry of sperm cells. Discussed that already. And C. Are drug companies developing a male contraceptive pill? Suggest 3 ways in which this could prevent pregnancy. Well, the 3 ways is this. So first, they could reduce testosterone levels, and testosterone is the hormone required for sperm formation, so that would reduce sperm formation. You could reduce sperm motility or delay or inhibit the formation of the flagellum, that's the tail of the sperm. So basically the sperm cells, the spermatozoa form, but they can't swim, so of course they cannot fertilize the egg. They could also thicken the mucus in the vas deferens, as the sperm duct, to prevent release of sperm cells into the semen. So what happens here is that since semen is released from the prostate and the seminal vesicles, that gets released but not the sperm cells, because it comes like the passageway gets blocked for them. Eventually the sperm cells in the testes would die. Okay, here we have June 2016, question 4. In the box below, draw a label diagram of the female reproductive system and using an arrow trace the path that spermatozoa, that's a sperm cell, would have to take to fertilize an ovum. You want to draw a diagram here that shows the vagina, the cervix that separates the vagina from the uterus. We have 2 oviducts or fallopian tubes, and then we have the ovary at the end. The passage for the sperm cell would be through the vagina, the cervix, the uterus, through the oviducts or the fallopian tubes, and then somewhere in the middle here it would meet the eggs. It does not go directly to the ovary, but somewhere in the fallopian tube it would come into contact with the egg cell and fertilize it. B. A young couple wants to choose between two contraceptive methods, barrier or surgical. Discuss why the male may prefer the barrier method rather than the surgical method of contraception. Your answer should include 2 advantages of the barrier method and 3 disadvantages of the surgical method. Barrier methods are basically condoms, and surgical methods are vasectomies, cutting of the sperm duct. So the barrier methods are temporary, which means you put on your condom, you take it off, you do your thing, you take it off. There's no long-term effects of it. And the man's reproductive ability is unaffected. There's no surgery or anything involved. There's no surgery, no health risks, and no post-operational recovery is required. Yeah, need nobody cutting open the scrotum to do the thing, you know what I mean? So the surgical method, or the vasectomy, involves the cutting and ligation, or the cutting and tying off of the sperm duct. It's more expensive, it removes all reproductive capabilities for the man, it's very difficult to reverse. The method provides no protection against the transmission of STIs, so whereas condoms do, because there's no exchange of vaginal fluids and semen. Reversal of a vasectomy is not likely in many cases. So we're looking at question 3. This is July 2021. Explain why a human embryo does not have large nutrient stores, but a plant embryo does. Human embryo basically will grow into a baby, remember that. So the human embryo is still attached to its parent. Remember, it has the placenta and the umbilical cord, so it receives nutrients via those extra embryonic membranes throughout gestation or pregnancy. It also helps to release waste through that umbilical cord, such as urea and carbon dioxide. Remember, the umbilical cord, if you really think of it, that was your first mouth, that was also your first anus, if you think about it. However, a plant embryo is found in a seed that would eventually be dispersed, and that will move away from its parent plant. So whether it's by wind or water or an animal, it will separate from its parent. So the parent don't just throw away the fetus while it's in it, it's still attached, but the seed would eventually separate. The plant embryo would require some kind of food storage, so endosperm or cotyledons to nourish the embryo. Those there would have enzymes that would break down large complex molecules into smaller molecules, and then you would have your cotyledons might go above ground, and then the true leaves would form to facilitate photosynthesis. So basically, the human embryo doesn't have nutritional storage or endosperm, and it is still attached to the parent. But the plant cell requires endosperm and is detached from the parent. And suggests 4 reasons why a couple would be advised to use the permanent method of birth control for the male rather than the permanent method of birth control for the female. Well, I just described that the one for the male is a vasectomy, and is a pretty non-invasive surgery. They just make a small incision in the scrotum, they numb the area, of course, and they cut a tiny piece out of the sperm duct and they tie off the ends. Basically prevents sperm cells from coming out when ejaculation occurs. The method for birth control for the female is a tubal ligation, and this is more expensive than a vasectomy. This is because you usually have to, you have to go to sleep. You require general anesthetic. It's a much more invasive surgery than a vasectomy. There's a higher risk of infection for tubal ligation if done inappropriately, and there's a longer recovery time because of the general anesthetic. Tubal ligation is a much more complex procedure and involves exposure to the abdominal organs because they actually have to, cut you open. Whereas vasectomies only deal with, a localized area, they only deal with the scrotum. So any 4 of these basically explain will give you 4 marks. And yeah, all this should be good. Good luck with your exam. What we gonna say? Good luck, good luck. We're looking for 1s, we're looking for 2s, we're looking for 3s, we're looking for 4s. Hey, just pass all your blasted exam now. Good luck all you.